Okay, so let us get started for today. I will start sharing my screen. Mhm. Okay. Hello everyone, welcome to the week 7 of the PMRF-NPTEL problem solving session on Introduction to Developmental Biology. Uh, I will quickly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow and PhD scholar at IIT Kanpur, and I'm taking these sessions. These sessions are aimed at solving previous year's quizzes of the same course so that we have a better understanding of the concepts as well as the how to answer the questions in the quizzes as well as the final exam for NPTEL. This week we are going to deal a lot about morphogens as well as morphogen gradients and syncytial development. If anyone has any questions regarding this week's course, then we can handle that first, or we can just go ahead with doing the today's session. Any questions by anyone?
No, no questions.
Okay, so let us get started. So, how we do these sessions is that I will show you a question from the previous year's run of the course, and I will ask you to try and answer the question, and then we will look at what the concepts behind the question are, and then answer it. So, let us get started for today. This is a pretty nice question on morphogen gradients. So, what does it say? So, imagine there being a morphogen gradient in a tissue. Imagine it going from left high to right low. And in one tissue it causes this kind of a pattern, you know, yellow followed by purple, then yellow, then purple, then yellow. In another tissue the same morphogen gradient from left high to right low forms this checkerboard pattern. Now the question is, if I transplanted part of the tissue from left top to the middle here and vice versa, so this goes here and this comes here, what will be the resulting pattern that we will see in the tissues? And these are the options right here, and these are different variations of what can happen, and the question asks you that what should happen if you just transplant these two around, making sure that the morphogen gradient remains the same. So you can put think about it and put in the answer as A, B, C, or D.
Actually, I don't see the options on
The
screen.
options are so this is one option, these two
Okay.
these two are one option,
Okay.
so this is essentially A,
Ah,
B,
yeah.
C, and D.
Okay. Can you explain it a bit more?
Yes, I can. Just give me a second. My Bluetooth doesn't seem to be working. Just give me a second. Am I better audible now? Okay. First, should be this. Yeah, good, excellent. Okay, coming back to this. So let us go through it a bit slowly. So morphogen gradients. So what are morphogens? Morphogens are signaling molecules whose concentration is what determines how the tissue will respond. So it was very well explained by Walpert, and we are going to discuss the model that he proposed, which is known as the French flag model. So I would like you to look just here. So imagine there being a source of the morphogen on the left end of this tissue, and since the source is here, the concentration is highest here, and then it drops down along the straight line all the way to here where it is the lowest. What the French flag model tells us is that when the concentration is very high, the tissue will respond with a particular morphogenesis. So say it will form the blue color. When you go a bit further, the gradient, the concentration will drop, and a medium concentration of the morphogen will cause a white colored change. So the cell will have a white color. And finally, when you go furthest away, then there is a very low amount of morphogen, and it will only give rise to a red color. So if you were very close, it will be a blue color, moderately far away white color, and furthest away it will have a red color. So you can imagine such a morphogen gradient will give rise to a French flag. So if my source is here, then the closest cells will be blue, followed by white, finally they will be red. Fine. So this is the first thing that you should know. Imagine there is a second tissue which has the same morphogen, but the cells respond differently, with the morphogen gradient being on the left hand side. So the highest on the left and least on the right, it actually gives rise to a tissue pattern which looks like the American flag. So same thing, closest cells respond to the highest amount of the morphogen and form a pattern like this, followed by something in the middle, finally a stripe pattern right here. Now what is the idea in, so now we are trying to understand if we transplant both of these tissues, what the final change would be. So what the experiment is similar to the question. You take part of the French flag here and transplant it to where it was in the American flag, and vice versa. So you have changed the tissues, but the morphogen gradient remains the same. Now the transplanted tissue itself, which has gone from the American flag to this position, receives the highest amount of morphogen now. But this particular tissue, which was taken from the American flag, responds to the highest amount of morphogen, not like the tissue in the French flag, but it will actually form the star pattern, because in the American flag the highest amount of morphogen actually causes the star pattern. So when I take a tissue from the American flag and place it on top of the French flag, it will actually form a star pattern right here. So imagine if the tissue was taken from anywhere in the American flag, giving it the highest amount of morphogen will cause a star pattern to occur. Same thing happens even if I replace it all the way in the French flag. It will form a star pattern, but just on top of the French flag. Similarly, if I now take part of the French flag and transplant it into the American flag, in this position you can see that the French flag actually has this corner here. So it will form part white and part red. And when I place it onto the American flag, it receives the particular amount of morphogen to form this pattern only, and that is what we see right here. So taking from the French flag, the tissues from the French flag say, yes, I am at a moderate amount of morphogen concentration, I should form a white and a red pattern. And that is what it forms. Because the tissues know only to respond like this, like how they have responded as the French flag. And that is why you have a pattern which is essentially this transplanted here. So to just reiterate, how it happens is that a tissue senses the morphogen concentration, which can be high, medium, or low, and then decides what it wants to form at that stage. So, for our French flag, anything in the middle would have been white. So, it will form a white color even though it is transplanted somewhere else. So we have seen experiments such as this, when we were doing experiments with the newt and the frog embryo, in which taking the newt suckers and placing them onto the frog actually developed suckers, or in case of the epidermis transplantation experiments, in which keeping the same epidermis over the thigh mesoderm or the wing mesoderm or the feet mesoderm gave rise to particular structures which are specific to the mesoderm from which it was taken. So the tissue can respond only in the way that it is already programmed to, and it will respond depending on the morphogen gradient. Is this fine? Is this okay? Okay. So coming back to the question, now let us look closely at what the situation here is. I am transplanting from this top corner to this middle region. First, so if I take this middle region and place it at the top left corner, here this tissue is now facing the highest amount of morphogen concentration. In the highest top left corner of the morphogen concentration, this tissue formed the checkerboard purple pattern, and that is what we see right here, because this tissue on the, if I placed on the top left, would have formed the purple pattern. So even transplanted to this particular color scheme, it still formed the purple pattern. Similarly, the tissue here, when transplanted to the middle, it actually formed this yellow kind of a line right here. So me putting it in the different tissue doesn't matter. It only thing that matters is the morphogen gradient. In the middle, here you can see it formed a yellow line. So even transplanted, it will form a yellow line right in the middle of this different tissue. So the correct option is this one. Is this fine? Okay. So that was one question on morphogens, and we now have a second one. So the question again is morphogen related. In this particular question, scientists are experimenting with the nodal protein to determine the effects of concentration gradient on embryonic development. The following diagram demonstrates the wild type embryonic development in the presence of a nodal gradient. So similar thing, on the left end you have the highest amount of nodal, which then drops down as you go to the right. Originally, the tissue responds something like this. So we have a morphogen secreting cell, and in the highest concentration, the first four cells, they give you a brown color. So this is obviously a protein which is being expressed by the highest amount of nodal being there. Now when you have a moderate concentration with the cells just a bit further away, you have a pink color, and in the furthest away from the nodal secreting cells, where the nodal concentration is the lowest, you have a blue color. So this is the scheme, similar to the French flag we saw. Now the colors are just brown, pink and blue. Now these people were doing a control experiment in which scientists removed some of the cells around the source cell and transplanted other cells which are represented by circles. So these circular cells right here. Importantly, these circular cells do not have a receptor for nodal, so they will not respond to nodal at all. Now the question is, what happens to the pattern when I insert circular cells which do not respond to nodal in the middle of all of this? So again you have four options: A, B, C, D, E, five options, and you have to choose which one should be the correct one when I replace circular cells in the middle. So what do you think will be how this pattern will change when I put in these circular cells which do not respond to the nodal signal? You can put whatever you think is the correct option into the chat, or just say it out loud. Okay. So option A. So let us just have a look at it, uh, at the what we are dealing with. So let me tell you a bit more about morphogens before we go into answering this question. So as I said, morphogens are signals which the cells respond to, and the differentiation of the response of the cells depends on the concentration of the morphogen. So people have done a lot of experiments to figure this entire model out. One of them is being described here, in which these couple of scientists, they took the same nodal mRNA and tried to figure out what the how the concentration gradient worked. So what they did is that they took an embryo and put in nodal mRNA plus a dye to figure out the concentration gradient onto a particular point in the embryo. So from a cross-sectional view, you see there, the lateral view, you can see that there is one point which has a lot of mRNA, and in the dorsal view you can see this is the point where nodal is being essentially, there is a lot of nodal, and this concentration drops down as you move further away. And if we just magnify this, this is what you will see. So these cells directly have nodal in them, followed by some more cells, and then you can see the closest ones show a red color, followed by white, followed by green, followed by purple, after which the nodal goes away. So essentially highest concentration here, lowest here, and you see that the color change depends on how much nodal morphogen they get. So the first experiment they started to do is that, let's see if it is indeed concentration based. So they put in a very low amount of nodal compared to the first experiment, and interestingly they saw low amount of nodal only gave them changes which is similar to what a low amount, a further distance away from a higher source would be. So cells all the way here, when they are 7 and 8, they receive a low amount of nodal. When you inject directly, you also see similar response. When they have a low amount of nodal, they only show the purple fade, which is should have been furthest away, because it is concentration dependent and not distance dependent. It is just that in the normal case, it is the distance which is equivalent to the concentration, because these cells receive the lowest amount. You inject a low amount and they show the similar change. Now if you put in like 10 times as much nodal, all the cells will show only the highest, the differentiation which the cells closest to the nodal signal do. That is because this is the highest concentration of nodal. So when you put in a lot of nodal, all of these cells now receive a lot of nodal, and so they will show only the change which the closest cells showed in the previous example. So all of them turned red because they receive a lot of nodal, and it just shows that it is concentration dependent. If you put in a lot of nodal, all of them will show the change as 1. If you put in a low amount of nodal, they will show the change as 7 and 8, because there is a low amount of nodal here. So this establishes that the morphogens work in a concentration gradient. Is this fine?
Yes.
Okay. So next, they figured out that in order to confirm that this is only a concentration dependent mechanism, they did a lot of control experiments. So they did a lot of negative controls. The first what they did is that, in this example they have taken cells which are circular as the ones which can respond to the nodal mRNA, and the box cells are the ones that cannot respond to the nodal mRNA. Now a question that they were trying to ask is that, say all of this does not depend on the concentration of nodal itself. It is where the cells in which you inject nodal, they undergo a particular change. They then do some kind of signaling, juxtacrine or paracrine signaling, which causes the further changes down the line. So how do we figure that out? That it is the nodal concentration doing it and not the cells responding to the nodal concentration, and they in turn causing all of these changes. So they did a lot of negative control experiments. The first they did is that they took a cell and then followed it by all cells which cannot respond to nodal. So they injected nodal mRNA in it, and they saw that this cell responded similar to a first experiment. It turned red, showing the highest concentration of nodal, but nothing else turned any other color, showing that okay, highest nodal causes red color and none of the other cells respond. So it must be nodal that is doing something. Next, what they did is that they took a bunch of cells which could not respond to nodal, followed by cells which could respond to nodal. They injected nodal mRNA here, and now the interesting thing is that if the hypothesis were correct that the first cell responds to nodal, in turn doing something to the others, which causes further differentiation, then this particular experiment should solve it. So the first cell could not respond to nodal. But even in that case, we saw morphogenesis in cells which could respond to nodal, which shows that it is not that the first cells produce a different signal. It is all about the nodal gradient, because the further away cells also respond as if they were receiving the, it is the nodal concentration which is causing the differentiation. Next, what they did is that they transplanted a particular cell from somewhere else which also could respond to nodal in the middle of this array of cells, and they saw when they did the same experiment, put in nodal in the first cell, this transplanted cell also acted similarly. It also became a green cell, which shows that it is the nodal gradient and the proximity to the concentration of nodal which is doing this. And finally, they put in lag Z mRNA to show that it is actually nodal doing it and not pricking by of the cells by anything. They put in lag Z and saw no morphogenesis. So all of these negative controls established that it is the nodal concentration from the source and the diffusion of the nodal and the gradient of the nodal which actually causes differential morphogenesis. So the cells receiving the highest amount formed red, followed by white, followed by green, followed by purple. This is how it was established. Now, let us go back to the particular question. So here the scenario is just flipped. It is the square cells or the rectangular cells which are responding, and the circular ones which do not respond. So we go back to this picture. So our yellow cell is the one that is producing the nodal signal. The closest one become red, further away become, so first four become brown or red, next four become pink, next four become blue. So if our circular cells could not respond, so they will not show any color change. So the first should not be the correct option, because they are showing some kind of color changes. uh,
Yes. Yes.
the second one, then you see that you put in two circular cells in between, but because they do not respond to the nodal at all, they remain white and do not affect the fourth cell from
Yes.
becoming brown. And then pink
Yes.
to white, pink to white, and then whatever blue. So the second option is the correct one, and the others are not. Perfect. So this was about morphogens. Now we go to a different kind of so this is about autonomous and conditional specification. So select the correct statement regarding embryonic development. Autonomous specification does not depend on interaction with the surrounding environment. Removal of cells from an autonomously specified embryo will result in embryonic lethality or an embryo missing certain parts. Conditionally specified embryos contain localized morphogenetic gradients. Developmental potential of individual cells in a conditionally specified embryo is higher than that of cells in an autonomously specified embryo. And cells of an embryo that follow regulative development can reach their fate regardless of the environment. So this is a more than correct kind of a question. So more than one options could be correct. So you can put in A, B, C, D, whatever you think are the correct options into the chat. Okay, Suba answered that the options are A, B, C, and E. A is autonomous specification does not depend on the interaction with the surrounding environment. Removal of cells from an autonomously specified embryo will result in embryonic lethality or an embryo missing certain parts. Conditionally specified embryos contain localized morphogenetic gradients, and E is cells of an embryo that follow regulative development can reach their fate regardless of the environment. Okay, so let us quickly go through the concepts behind it and get back to the answers. So what is autonomous specification? So it is also known as it causes mosaic development. So different regions of a egg cytoplasm contain different localized morphogenetic determinants. So all the cells after one stage are predetermined to form particular structures in the embryo. So they have already been determined in their fate specification. So a good example is normal development of a patella, which is a kind sea organism, which is like barnacles. So in the developing embryo, if the embryo is just allowed to grow, then the cells marked with yellow will form these uh, hair-like projections, which the organism uses to sense its surroundings. Now, if you take part of this presumptive trochoblast out of it, which was to form these hair-like projections, and let it grow in isolation, it can still give rise to the entire structure. So, autonomous specification, like it doesn't care where it is, in isolation, in adjacency to a different kind of cell, in a different embryo, it knows what it is going to form and it is going to form it whatever you're going to do. So, it is known as autonomous specification. So these kind of autonomous specifications, what they cause is that if cells are now removed from this embryo, say when I remove the presumptive trochoblast from it, this particular structure, which would have been likely this one, does not form in the final embryo because all the cells were predetermined to do what they must. And once you remove cells from such an embryo, it will not form that particular structure. So that is what autonomous specification is all about. Cells know what they are going to form and they are just going to form that. If the embryo is missing some of the cells, so it is missing some of the final embryonic structures because those cells cannot be replaced by any other cell. So this is autonomous specification. Next, we talk about conditional specification, which gives rise to regulative development. So previously it was mosaic development, now it is regulative development, in which interaction with neighbors regulate the development. So the cells actually listen to their local surroundings and form structures depending on that. So for example, in a zebrafish embryo which undergoes conditional specification, this can be easily shown by taking some of the cells from one part of the embryo and replacing it to another part. So cells from here go to this part and cells from here go to this part. There should have been a reverse arrow here as well. And we see that instead of forming the gill-like structure that should have formed from this part, the cells form a normal fin, as if they listened to the cells surrounding it and actually agreed that okay, I now have to make a fin. Even though I was supposed to make a gill, now I am surrounded by cells which are asking me to make a fin, so I will form that. Similarly, cells which were supposed to normally make a fin, when transplanted here, they form the gill-like structures because they listened to the surrounding cells and were then asked to form a gill, and it formed a gill. So, conditional specification, in which cells are not predetermined to form what they are going to form. They can be influenced and they can change their fate depending on where you exactly put it. So most higher organisms undergo a conditional specification where this some of the cells, if you transplant, they can actually form other structures, and this also helps in the case where some of the cells die, or some of the cells just apoptosize out of nowhere, or some of the cells are removed. Other cells can take their place and form the structures instead. So it is not like if you take remove some of the cells, the entire embryo will be losing embryonic structures further down development. This entire embryo will ask other cells to take their place. So conditional specification. So interestingly, in these examples, we have looked at particular embryos which undergo autonomous specification and which undergo conditional specification. But in most embryos, both of these go hand in hand. Some structures at some stages undergo autonomous specification. Some structures at some level, some developmental stage undergo conditional specification. It is not either or, but both of them together. Okay, now coming back to the options, let us go through them depending taking back what we learned. So the first option is correct. Autonomous specification does not depend on the interaction with the surrounding environment, which is true. Cells do not care at all about what the surrounding environment is. Removal of cells autonomously specified embryo will result in embryonic lethality of the embryo missing certain parts. Absolutely correct. Conditionally specified embryos contain localized morphogenetic gradients. This one is not correct because autonomously specified embryos actually have localized morphogenetic gradients based on which they form particular structures. Conditional ones actually depend on the global morphogenetic gradients, and that is why that is the signal that they're listening to from their surrounding or locality. So these guys depend on a global morphogenetic gradient instead of a localized one. Developmental potential of an individual cell in a conditionally specified embryo is higher than that of the cells in an autonomously specified embryo. This one is also true, which we saw from this example right here. In a conditionally specified embryo, cells from a different part can form a structure which it was not supposed to form previously. So it is much more developmentally elastic. So it can form different structures depending on where you put it. So its developmental potential is much higher than an autonomously specified embryo, in which the cells know what they are going to form and they cannot form anything else. They are only going to form what they want. And finally, cells of an embryo that follow regulative development can reach their fate regardless of the environment. This is not true because regulative development means conditional specification. So if I took these cells and put them in a different environment, say I blasted it with a nodal morphogen, then they are never going to form the structure they were supposed to, because they depend on the environment to form particular structures. They listen to the surroundings and the locality. Instead, autonomous specification, it doesn't care. You put in nodal, BMP, FGF beta, does not care. It is going to form what it was supposed to form. So this is also not the correct option. So the correct options were A, B, and D. Okay. Any questions? Okay. Moving on. Choose the correct option which correctly shows the matching pairs. So here we have a developing Drosophila oocyte, and there are multiple different cells here, and we are asked what does these A, B, and C specify. So A, B, and C are the markings, and 1, 2, 3 are going to be the particular cell they identify. So nerve cell, oocyte, and follicle cell. So we have essentially a match the following kind of a thing. So you have to choose A being what kind of cell, B being what kind of cell, C being what kind of cell. So you can choose between A, B, C, D, and E, and put in whatever you think is the correct option to the chat.
I can see the image clearly, and I don't know where is A, B, and C as
Yeah,
well.
let me zoom in, and can you
Yeah,
see it now?
yeah. Now I can see a faint image.
Okay. So the options are nerve cell, oocyte, and follicle cells. So I'll just shorten it on a bit, and yes, now we can cover both of them. So we have the options just here. Yes. Okay. So C is follicle cells, B is oocyte, and A is nerve cell. You're absolutely correct, but let us just quickly go through. Uh, yeah, you are absolutely correct. A is nerve cell, B is oocyte, and C is follicle cell. But we quickly go through a bit of Drosophila oocyte development, because we have a lot of questions lined up depending on this after this. So the idea behind why are we even interested in the Drosophila oocyte, we actually want to learn something known as syncytial development. So we have seen regulative development, which occurs due to conditional specification. We have seen mosaic development, and now we are looking at something known as the syncytial development, which occurs very nicely in the Drosophila oocyte, or the developing oocyte, or the Drosophila embryo. So that is why we are trying to now understand Drosophila embryos, and how the oocyte develops. So this is a Drosophila fly, and in the abdomen there is a particular structure which is called the ovary, where all the oocytes are being made. And if you take a very close zoom into the oocyte, then you can look and see at what the germarium is. This is the primordial oocyte, where actually everything is starting off. So at the end of the germanium, there at one end there is a signal from the ovariole, which is DPP, which is a TGF beta ligand, which signals to the first two cells. This is known as a germ cell niche. So here the germ cells are producing new cells all the time, and they are, and they produce it by actually receiving the DPP signal. So germ cells being cells which divide, or stem cells which so this is a stem cell, a germ stem cell niche. So these stem cells will give rise to germ cells. So how does it happen? The signal from DPP is directly close to the stem cells. So the stem cell divides and the division occurs longitudinally. So this is one cell, a new daughter, this is a second daughter. And once this happens, the second daughter, which is now called the cystoblast, and further away from the DPP signal, so that it does not receive the DPP signal because it is right here, it no longer acts as the stem cell. So this daughter is a result of an asymmetric division. So one cell remains as the germ stem cell, the other becomes cystoblast. This cystoblast will give rise to the entire oocyte. So this cystoblast undergoes a lot of divisions in a very interesting manner. So this is a cystoblast which was one, and now it starts dividing. Once it starts dividing, it under it undergoes incomplete cytokinesis. What that means is that the cell membrane doesn't separate. So once the first cystoblast cystocyte divides, it forms two cells, but they are connected, and they are connected by a ring canal. So this is known as a ring canal. So essentially, if you do not separate the cell membrane into two, so obviously the cells are going to be connected. So here is where the connection is. The cells continue to divide and they maintain this kind of incomplete cytokinesis, so that more and more cells, when they come off, they maintain a connection to the original two cells. So 1 now divided into 1 and 3, and 3 is connected to 1, and 2 divided into 2 and 4, and both of them are connected, but 3 and 4 are not connected because they were never in contact. This is the cell that budded out and formed another cell. So this is where the connection is: 1 connected to 3 and 2, 2 connected to 1 and 4. This continues till you have 16 cells which are all connected by these ring canals. But here you can see that only the original 1 and 2 are connected to most of the cells. So 1 is connected to 1, 2, 3 and 4. 2 is similarly connected to 1, 2, 3, 4 cells, and every other cell is connected to lesser and lesser cells. So for example, 14 is connected only to 6, 6 is connected to 2 and 40. So essentially all of these 16 cells have all of the translational or transcriptional output. So all the proteins they are forming, all the mRNA they are forming, all are connected to these 1 and 2, and essentially these cells are feeding all their transcriptional and translational output to these two cells. Now one of these two cells will become the oocyte. So the oocyte, you obviously would know, is a much bigger cell, and it is bigger because it is feeding of essentially all of these other cells, and all of these cells are known as the nurse cells, and they essentially nurse the oocyte, which becomes bigger and bigger and ends up just surviving all of these nurse cells. And looking at them in the oogonium or the germarium, you see, so this is the stage where they have all divided, but the oocyte is not specified yet. Now the oocyte is specified, and it is growing larger and larger because of the cytoplasmic input from the nurse cells. And here is where we end up, where this is the oocyte, which is huge, and all the nerve cells are still connected to the oocyte and pushing the cytoplasmic input into this particular oocyte. So there is one point that I would like to specify here. This nerve cells and the oocyte all came down as descendant of the germ stem cell. So these are of a germline lineage. All of these nerve cells plus the oocyte. But there is a layer of other cells which covers the entire egg chamber. These cells are known as follicle cells, and these are somatic in origin. So these did not come from the germline lineage. These came from the somatic lineage. So mother's own cells which are different from the gonads or any cells around it. So follicle cells, somatic origin, germline cells give rise to oocytes and the nurse cells. And here are some nice pictures of them. Okay, now looking just at the egg chamber, this is our oocyte, these are our nerve cells, and here all around it is the follicle cell layer. And here is a tease about what we are going to learn next about how the axis specification occurs. We will get back to this. But for this question, this is what we needed. These are all nerve cells, this is the oocyte, and this is the follicle cells. Yes, finally coming back to the question, you were absolutely correct, Suba. It is A is nerve cell, B is oocyte, and C is follicles. Is this fine? We move on. Okay. Now, we actually delve deeper into how axis specification occurs in the oocyte. And the first question that we are asked is, becoid mRNA moves to the anterior with the help of dash motor proteins, and nanos mRNA moves to the posterior with the help of dash motor proteins. So fill in the blanks. What is the first one? What is the second one? And just as a hint, uh, becoid moves towards the anterior end, which is the negative end, towards the center of the cell, and nanos moves to the posterior end, which is the positive end. So if you know which motor goes to the negative and which motor goes to the positive, you can easily answer this. Any ideas? Okay. So there are two protein motors which are dynein and kinesin. Dynein moves to the negative end or towards the center of the cell, and kinesin moves to the positive end or to the periphery of the cell. Becoid moves to the center and anterior. So So first is dynein, second is kinesin. But since we have the chance, we will actually look at anterior posterior axis specification based on this question, and rather not keep it at dynein and kinesin. Okay, so here was we were right here, right? So I will go 2 slides back. Okay, so even at this stage when the oocyte is not fertilized into a zygote, already anterior posterior specification has starting to occur, so to break the symmetry. How does it happen? There are uh, so all these nerve cells, they produce a particular protein known as gherkin. Gherkin right here, these are these green dots. By a now unknown mechanism, these gherkin proteins are localized to the posterior end of the oocyte, and the follicle cells that we are talking about, the somatic origin cells, all have receptors for gherkin known as torpedo. So obviously the posterior most cells now receive the gherkin signal because gherkin is localized here, and they differentiate into terminal follicle cells. That is the first point at which actual symmetry breaking is occurring and anterior posterior is being divided. So here is where we start the story. Gherkin being attached to terminal follicle cells, and these guys have now differentiated. Now once they are differentiated, these cells actually send back a signal to the oocyte. This signal actually causes formation of polarized microtubules. So it causes positive ended microtubules to form here and negative end here. So when we are talking about microtubules, microtubules are actually these long, you can imagine them as ropes that travel all across the cell, and these actually are made by addition of more and more monomers to the already growing chain. So essentially adding one link at a time till you have a big rope. So one end is where the entire chain starts, which is known as the negative end, and the other end is where you keep on adding more and more stuff so that it becomes longer, and that is the positive end. So these terminal follicle cells cause the microtubules to elongate in their direction, actually giving rise to a negative on this side and a positive on this side. So once you have a negative end and a positive end determined, now the cell already has the cytoplasm already has proteins which are known as microtubule motors, and these have specific directions. So dynein motors always travel towards the negative end, and kinesin motors always travel to the positive end. And what happens is that there is a particular mRNA which was deposited into the oocyte, which was first all over the place. But this bicoid mRNA actually can latch onto dyneins and is now deposited on one end, and another protein known as Oscar, as well as there are multiple proteins like nanos as well, which latch on to kinesins are and are localized to the positive end, essentially giving rise to the anterior-posterior identity of the oocyte. Because bicoid is a morphogen which determines the anterior end, and Oscar and nanos are morphogens which determine, Oscar is not, nanos is the morphogen which determines the posterior end. So essentially, with the Gherkin signal followed by terminal differentiation of this follicle cells, followed by formation of polarized microtubules, and finally differential localization of morphogens, we establish the anterior posterior polarity of this oocyte. And now, because of stuff that the bicoid protein as well as the nanos protein do, we have anterior posterior specification. So this part will only be anterior and this part will only be posterior, which we will learn in later lectures. But yeah, this is how entire thing starts off. Is this fine? Okay. So here you can see with the in situ hybridization for bicoid mRNA, you see that it is only localized to the anterior end, and a nanos is localized to the posterior end, which is not shown here. And another interesting thing that happens is that once this polarized microtubule network is established, the nucleus itself also starts moving. It latches on to dynein motors and moves all the way to the dorsal anterior. So dorsal is essentially, for this it is the top end. It moves to the top and anterior end, so it moves all the way here, and it also carries a lot of gherkin protein all around it like a halo, which in turn gives rise to the dorsal ventral polarity. So till now we have established anterior posterior, but we still have not established dorsal ventral in the entire oocyte. That happens after the nucleus travels to the dorsal anterior portion, which we will see in a bit. Okay, so coming back to the question, bicoid moves to the anterior with dynein and nanos moves to the posterior with kinesin. This one is, the question was pretty easy. Okay, straightforward. Okay. So question, this question is based on people actually trying to figure out where the torpedo receptor should be present for correct axis specification. So is it that the germline expresses torpedo, or is it that the somatic cells expressing torpedo which actually causes the final proper axis specification? So what did they do? They took a particular embryo which was wild type, so it has everything normal, it has torpedo expressing, and they took and transplanted the pole cells. So the pole cells are the one what will form the germ cells later on from these particular embryo, and transplanted it into a different embryo which was completely torpedo deficient, so it did not have torpedo at all. So the germ cells were placed here, and the pole cells from the torpedo deficient embryo was placed onto the wild type uh, embryo. So essentially I am putting, so now both of these embryos are mosaic in a way that germ cells of this particular embryo does not have torpedo, but everything else has torpedo, and germ cells of this embryo has torpedo but nothing else has torpedo. So now the question is, uh, which of the following? So embryo B goes to form a normal dorsal ventral axis while embryo A does not. Embryo A goes to form a normal dorsal ventral axis while embryo B does not. Torpedo is produced in the follicle cells, torpedo produced in the germ cells. Embryo without proper axis formation will be ventralized. Embryo without a proper axis formation will be dorsalized. So here we have just seen the role of torpedo in anterior posterior axis specification. It also has a role in dorsal ventral axis specification with similar signaling from Gherkin, but the downstream is different, which we will look at. But here the question is pretty straightforward. Which type of cells need to express torpedo for correct axis specification? And this is the particular experiment that they did to figure it out. So this is more than one correct option kind of a question. So you can choose more than one, and you can put what you think is the correct option in the chat. Just give me a second. Okay, so we got the option B, C, and E, which is embryo A goes on to form a normal dorsal ventral axis, while embryo B does not. The torpedo protein is produced in the follicle cells, and E would be the embryo without proper axis formation would be ventralized. Okay, let us look at it. Okay, so we were so far in the story that we have anterior posterior specified, and now we are going to look at how dorsal and ventral is specified. So the first thing that happens, as I said, that the nucleus along with the halo of Gherkin proteins travels to the dorsal anterior. This is the initiation for dorsal ventral axis specification. So here you can see when you do a mRNA hybridization for gherkin, it actually is localized to the dorsal anterior as we saw, and here you can see it in a cross section, and you see yes, particularly in the dorsal. And here is a antibody staining for the same, which says the same thing. Okay, so this is the experiment that we did. We did a germline chimera, took the wild type pole cells, placed them on the torpedo deficient embryo, and took torpedo deficient pole cells, placed them on the wild type embryo. And now you want to see what happens. Okay, so first thing that happens is that both of these embryos will grow up to produce a wild type, a normal female, but the first fly will have all the somatic cells having torpedo, but the germ cells, the ones that give rise to our nerve cells and the oocyte, not having torpedo. So this is what the embryo from the next generation would look like. So the germ cells not having torpedo, everything else, especially the follicle cells, having torpedo. And if you remember, the follicle cells actually are the ones which have the receptors for torpedo, and they receive the Gherkin signal. So it does not matter if the germ cells have torpedo or not. It matters if the uh, follicle cells have torpedo or not. So the torpedo deficient oocyte in wild type follicle will give normal dorsal ventral axis, because all depends on the follicle cells having torpedo. In the other case, wild type cells in a torpedo deficient female, the opposite scenario, the oocyte and the nerve cells have all the torpedo they will ever need, but the follicle cells do not. And the torpedo in the follicle cells is what matters, because they are the ones that receive the signal. And since they cannot receive the signal because there is no torpedo around, no dorsal ventral axis formation, and the entire embryo would be ventral. So this is what it is. We will look at this in the next question. What happens here? Okay. Yeah, we'll look at this in the next question. Okay. Which of the following statements are true regarding the experiment? The correct options are: the torpedo protein is produced in the follicle cells, yes, and the embryo without proper axis formation will be ventralized. This is what we will see how this happens in the next one. Is this fine?
Yes.
Okay. Just one more thing. The first two options are not correct. Reason being, uh, see, this embryo is already
Mm.
formed, and this embryo is already also formed. The pole cells will form the egg chamber for the next generation. So both these A and B embryos will give rise to perfectly viable adults. So the option say embryo B will have normal dorsal ventral axis, but embryo A will not. But that is not true. Both of these will form normal adults, just one of them will be sterile. It is not that these embryos are affected, the next generation is
Yeah.
affected.
Yeah. Yeah.
Perfect.
Yes. Yes.
Okay. Okay, so this we now look exactly at how the dorsal ventral specification works. Which of the following statements regarding the toll receptor are correct? Toll is present on the follicle cells, receives signal from the oocyte in the form of noodle protein. Toll binds to spatzula, or triggering the release of dorsal previously bound to cactus. A truncated version of toll receptor would result in constitutive degradation of dorsal cactus complex, causing ventralization of the embryo. A truncated version of the toll receptor would not be able to activate tube and pili, resulting in dorsalization of the cell. A loss of function mutation in the toll receptor can be rescued by blocking the release of pipe protein from follicle cells. This is also more than one option correct kind of a question, so I will let you try and answer this if you have an idea. Okay, tall is present on the follicle cells, receives signal from the oocyte in the form of nudal protein. That is correct. But this is a more than one option kind of a question, so there are multiple here. But let us look at the cascade and then come back to this. So, in our story so far, our nucleus with the halo of Gherkin mRNA and proteins has traveled, Gherkin mRNA has traveled to the dorsal anterior portion. So now this causes the entire embryo to now have a dorsal ventral axis. How does it function? So Gherkin mRNA here is translated to Gherkin protein, and it does the same thing. It goes to the follicle cells close to it and causes them to differentiate. And obviously the torpedo is required in the follicle cells to do this, and essentially the dorsal follicle cells are now uh, actually differentiate and do not interestingly have any signaling. So essentially gherkin at this point going to the dorsal follicle cells causes them to stop a particular type of signaling. What is that type of signaling? That happens in the ventral side because there is no gherkin here, and thus there is a particular uh, these ventral follicle cells which did not receive the gherkin mRNA or gherkin signal produce a particular protein known as pipe. Pipe goes ahead and is secreted to the perivitrilline space, which is the space between the follicle cells and the oocyte. And for these ventral follicle cells secrete pipe into this space. Now pipe gets activated by a different protein known as nudal, which then goes ahead and cleaves different proteins in the entire perivitrilline space. So pipe goes and cleaves GD or gastrulation defective. Once GD is cleaved, it also becomes a protease and cleaves the next protein in line, which is snake, which gets cleaved and becomes an active protease. The activated snake then goes and cleaves ester, which then becomes activated protease, and activated ester goes and finally cleaves spadzla, which then becomes a signaling molecule. So essentially similar, this is a signal transduction cascade, but instead of someone phosphorylating the next person, they are just cleaving the next person down the lane. So what happened, coming back to it, the dorsal cells receive a gherkin signal. They do not synthesize pipe. The ventral cells receive no such signal, so they produce pipe and secrete it. Pipe gets activated by nudal and then starts cleaving the next partner, which is GD. GD cleaves snake, snake cleaves ester, ester cleaves spadzla, which finally forms the uh, activated spadzla signal. So this is a signaling ligand which binds to toll receptors on the oocyte. Once toll receptor is bound on the oocyte, it activates pele and tube, which now does something interesting. So here in the now by this stage, the oocyte is already fertilized, and it has formed the syncytial egg chamber or the syncytial embryo, in which there are multiple nucleuses going around, so multiple nuclei all around, but all of them share the same cytoplasm. In the cytoplasm there are two proteins, dorsal and cactus, where cactus holds on to dorsal and does not let it go into the nucleus. But all of these pipe signaling going through the spadzla and toll actually degrade cactus so that dorsal can go into the nucleus, and dorsal going into the nucleus is the ventralizing signal. So this causes all the nuclei which were close to the ventral region actually to now receive the ventralizing signal, which is dorsal, and ventralize that part of the embryo. So essentially This entire region, which did not receive the initial Gherkin signal, will become ventralized, and that is how you form the dorsal ventral polarity. So nuclei here on the top here, they will produce particular signals which will cause dorsalization from this part, and ventralizing signal from here will cause stop of those dorsalizing signals and activation of different genes which will cause the embryo to get dorsal signals right here, which we will also look at in the what these signals are. But essentially all of these started with the Gherkin signal not being received in the ventral side, pipe being synthesized, the entire cascade, dorsal going into the nucleus, causing ventralization of the embryo, or the ventral end, ventralization of this part of the embryo. Is this fine? Okay. Which of the following are true? Toll is present on the follicle cells, receive signal from the oocyte to in the form of noodle protein. That is not true. Toll is present on the oocyte itself. Toll binds spatula, triggering the release of dorsal, previously bound to characters. True. Truncated version of toll receptor would not be able to activate tube and pele, resulting in dorsalization of the cell. That is true, because this toll activating tube and pele actually was ventralizing this part of the embryo. So without any functional toll, truncated toll, entire thing will be dorsalized. Okay, moving on. The presence of blank ligand differentiates between the mesoderm and the mesoectoderm. So I guess I have to show you this because this is I don't. Do you have a clue which it is going to be? What is the ligand that differentiate between the mesoderm and the mesoectoderm? Yes, I imagine. So yes, we shall look at this. Okay. So you remember, so our ventralize, now we are looking at what the ventralizing signal is, what dorsal does. So dorsal, which is the protein dorsal, which is actually the ventralizing signal. So it gets a bit confusing. But dorsal is the name of the protein which ventralizes the embryo. So it does a lot of things. First thing it does is that it inhibits all the genes which are required to form dorsal structures. So DPP and everything else which form dorsal structures, that are inhibited. So the dorsal structures such as the amnioserosa, dorsal ectoderm, lateral ectoderm are not formed here in the ventral side because dorsal is inhibiting all of these genes. Next, it activates three transcription factors: twist, snail, and rhomboid. Twist, so here you can see, so this essentially causes twist and snail, as well as both of them together first determine this to be the mesoderm layer, and twist in turn activates snail further, but snail inhibits rhomboid. So in the closest region, so in the most ventral region, rhomboid is not activated, and rhomboid instead is activated a bit further down in the neurogenic ectoderm. And snail also inhibits FGF8 ligands, so there is no FGF signaling going on in this mesoderm, whereas twist activates FGF8 receptor. So there are receptors all over this place, and once you go further down this channel, you have FGF protein forming, and which in turn gives rise to mesoectoderm. So this is too complicated. The chart is much easier to see. So essentially, uh, twist, snail, both of them specify the mesoderm. Rhomboid specifies the neurogenic ectoderm, or the ectoderm that will form neurons. FGF8 helps in gastrulation movement of the mesoderm, as well as differentiates the boundary between the mesoderm and the mesoectoderm, which will form the glia. And There are multiple other genes such as Tinman, which helps in heart development, DMET2 muscle differentiation, backpipe visceral muscle development. So too complicated. You just have to know these the following things. Dorsal is a ventralizing signal which activates twist, snail, and rhomboid. Twist and snail in turn activate downstream partners, including FGF8 receptors, which are activated by twist, which causes the boundary between the mesoderm and the mesoectoderm. Snail inhibits rhomboid, causing it to be expressed further in the neurogenic ectoderm. Twist gives activates further downstream partners such as Tinman, which form different parts of the mesoderm, which is the heart, the muscle, and the visceral muscle development. So here the question that was asked was presence of what ligand differentiate between the mesoderm and the mesoectoderm. It is the FGF8 ligand, just coming from this particular chart. Okay, so this I think is the last question for today. Match the following proteins involved in axis development and their function. DPP, Tinman, Backpipe, Snail, and FGF8. This is just what we saw. This is just in match the following thing. Visceral muscle development promotes gastrulation in mesoderm, blockage of neuroectoderm genes, promotes dorsal structures, heart development. So, if you remember this, or I can just show it to you, that this is just the previous chart as a match the following. So we saw, yes, you can try. Please go ahead. Okay, it is one is D, decapitated, which is DPP, promotes dorsal structures, which is correct. Two is E, Tinman is useful heart development, absolutely right. Three is A, backpipe, helps in visceral muscle development, perfect. Four, snail is C, blockage of neuroectoderm genes, which is correct. Five is B, which is promotes gastrulation of mesoderm. So all of these gives us which option? 1D and 2E. So this is this option, which should be D. I think that is also the correct choice. Absolutely right. Okay, perfect. Okay, so this actually brings us to the end of today's class. So we have learned a lot. We have gone through morphogen gradients, different kinds of autonomous conditional specification as well as syncytial specification, and gone through how axis development in drosophila embryo works. So any questions from today's session?
Uh, Not so far, but it was a good session. You explained it very well. Thank you very much.
Thanks a lot for attending as well. So okay, so if there are no questions, then we can end today's session, and thank you for joining today, and I will see you in the next week's session. Okay,
So,
thank you.
okay.